I want to thank the organizers, of course, for inviting me to this very cool meeting. And I think it's a little distracting that we have a spectacular view for the speaker, but you guys just have to look up front. But you'll all get your turn. Anyway, I think this is a great start to the meeting. Our first talk came from a mathematical perspective, and this talk is going to be very much from a biological perspective. I'm an experimental biologist, and so the way that I think about understanding sort of underlying properties in the mathematical sense that we all share in this room is usually prompted by observations of cell behaviors. I'm particularly trying to find things that are not easily explained by sort of standard models of cell-to-cell variation. And so today I'm specifically going to talk about cell migration as a test case for places where we can do quantitative measurements in a way that I think will fit in very nicely to the way that people are thinking about cell states and cell state transitions. For the specific experimental data that I'm going to show today, almost all of it was done by a very talented graduate student, Hansu Kim. All right, so we just got a great introduction to the concept of cell state as defined by a vector of all of the different gene expression levels. As you all know, and as came up in a question, that's only one little aspect of cell state. And when I think about cell state, or many of my fellow cell biologists think about cell state, we know already that just knowing RNA expression profiles is not going to be sufficient. Ideally, we'd want to know many other things that could be measurable about cell states in order to try to put this into a more rigorous formulation. And this includes not only things like protein levels, post-translational modifications, all of those things together with RNA expression, we would put into the category of molecular census, so just describing what's inside of the cell. At the same time, there's very good quantitative metrics for a lot of other aspects that determine cell state and cell fate, including things like organization, so where are things distributed within a cell, cell function, so is something secreted, is something motile, and also the environment, of course, influences the cell state. And overall, the hypothesis that Susan Rafalski and I together developed in a perspective we published a couple of years ago is that in a stable cell state, the stability of that state is determined because there are multiple two-way interactions among all of these different aspects of measurable things that are associated with a cell. And so to really try to understand cell states and why they're stable, it's good to think about all of these things simultaneously. So for today, I'm specifically going to focus on function and also on heterogeneity. So even within a population of cells that are sensibly the same, meaning they're the same cell type in an animal, or for example, they're the same cell line in an incubator. We sort of expect that they're going to share certain aspects of their cell state, but we know all of these measurements are heterogeneous across cells. We're all very used to thinking about heterogeneity in terms of molecular measurements, but I particularly want to describe heterogeneity through a primary lens of cell function, rather than through any of the other metrics that we might be more familiar with in molecular census or in cell organization. So for me, cell migration is a particularly good example of kind of cell function, which can be characterized quantitatively extremely well. In particular, my group focuses on actin-based cell migration, which is illustrated here by two wildly different cell types from two different animals. On the left, we have a fish skin cell migrating in culture and time-lapse, and on the right, that's a human neutrophil actually migrating in real time. So although these cells look very different and they actually have wildly different biological functions, they actually share the same underlying mechanism for cell motility, which involves assembly of the actin network at the leading edge, formation of adhesions to the substrate so they can pull themselves forward, and then contraction and disassembly at the cell rear. And one of the reasons that I think this is a good test bed for this sort of thinking is because we know already from many decades of detailed biochemical work that every single force-generating module in the cell, so for example, the focal adhesions that enable cells to contact their substrates, those have tens or hundreds of proteins that all have to work together. And you can disrupt many different aspects of cell motility by upregulating or downregulating or mutating hundreds or thousands of different factors in the cell. So in thinking about complex emergent behaviors, this seems like a good test bed. Today, we're going to specifically talk about cell motility from the perspective of cell migration speed. And it's been known for a long time that if you look at cells migrating around, for example, inside of animal tissues, that there's a huge amount of heterogeneity in just the speed at which they move. So the simplest possible measurement you can make about a moving cell, how fast is it going, you'll see a big range. Now, when you look at this inside of an animal, you don't really know whether it's because the cells are intrinsically different from each other or whether it's just because they're crawling through a very complicated environment, and so their reaction to the environment might be different. But the bulk of the evidence suggests that this is really cell-to-cell intrinsic differences. And I just want to cite this one really nice recent paper that's on bioRxiv looking at primary T cells. So these are cells isolated from a mouse. They're fully differentiated, totally normal cells. And the experiment here is to actually put them in vitro and make them run on a little track. So it's just a single cell running on a track. The tracks are all identical. And there's nothing for them to collide with. There's no variation in the environment. But even so, you still have this massive distribution in terms of individual cells going at a wide range of individual different speeds over a couple orders of magnitude of speed variation. Now, in my group, we frequently make use of this EL4 lymphoma cell line. This is a T cell derived cell line from a mouse, so it's immortalized. It lives in the incubator. We can do a lot of molecular manipulations to it. And this also demonstrates the same sort of speed heterogeneity that's also been shown in real T cells. So the experiment that I want to describe is it was actually originally designed as sort of a control experiment for a different project, but the control ended up being so interesting that now this is what we ended up studying. So we wanted to know if we could physically separate the cells that are moving fast from the cells that are moving slow. And for experimental convenience, we use this assay where we take our lymphoma cells and we put them on top of a track etch membrane. And this membrane is a very thin piece of plastic that's been bombarded with heavy ions. So it's got a bunch of holes drilled through it, and all of the holes are exactly the same size. So for these experiments, it's all going to be EL4 cells crawling through 8-micron holes, illustrated over here, drawn to scale. So to assay, to separate cells that are fast moving from cells that are slow moving, what we can do is what's called a transwell assay, where we put the cells above one of these membranes in tissue culture, and then we put a chemoattractant below them. In this case, it's a cytokine that T cells like called SDF1 alpha. And then over time, the cells will migrate through the holes and end up in the bottom of the well. And then you can just capture those cells and analyze them or propagate them or whatever you like. And so Hansu started off doing a series of selection experiments where he would do that transwell assay and then capture the cells that came through, take those cells, proliferate them up, and then put them through the same assay again, and then do this over and over again. So we can do essentially rounds of selection to try to find the fastest moving cells in a population over time. Now, initially, as you'll see, only a very small number of cells come through in the first round, and so we have to actually grow them up quite a bit. And in tissue culture, forcing cells to proliferate a lot is also a form of selective pressure. And so the control that we did essentially was to count how many cells came through the membrane each time, and then take that same number of cells from a population that had never seen the membrane and proliferate them. So we have matched proliferation burden, although only one of those two populations was forced to migrate. Yeah.
Maybe a stupid question, but how do you separate healthy versus unhealthy cells? Because that might be correlating with the migration.
We don't do anything about healthy versus unhealthy cells. These are, grown in tissue culture, and basically we just put them directly on the membrane. And so if there were a subset of cells that were dead or that weren't healthy, they wouldn't crawl through. So we wouldn't see that.
But through multiple rounds, do they get damaged? And if the faster one...
When I say multiple rounds, after they come through the first time, they have to grow up, between 5 and 10 generations. So it's a massive proliferation. So when you say healthy, that can also be defined many different ways. One way it could be defined is, can they continue to proliferate, right? So a dead cell, for example, is not going to proliferate, or one that has some sort of fatal flaw with its energy balance. So everybody we're looking at here is capable of proliferation by definition. Yep.
Do you have an estimate of clonality, or like how?
Let me come back to that later. Yep.
How big are the cells and how big are their nuclei?
So the cells themselves are about 20 microns across. The nuclei are fairly big, so they're sort of 10 or 12 microns. So crawling through these 8-micron holes. Sorry?
Challenge to get through for the nuclei.
It's a little bit of a challenge, yeah. And if we use a smaller hole, like if we use a 5-micron hole, we get many fewer cells through. So they're just at the point where they do have to squeeze a little bit to get through. Yeah.
So Julie, how much is cell diversity versus the cells check what their neighbors are doing?
Well, that's a great question. I'm not going to answer it, but I will show you how you can get there. All right. Okay, so that's the experiment. Here's the result. So this is the number of cells that come through the membrane after 4 hours of migration. And as you see in the first round, it's only a couple of % that come through. But if we catch them, let them proliferate, catch them again, let them proliferate, catch them again, let them proliferate, after about 6 rounds of selection, we can get about half the cells coming through the membrane, which is a very significant difference compared to what the population was to start with. Now, this is an immortalized cell line that came from a mouse that had actually been, experimentally given a tumor. They've been propagated in tissue culture for decades. And so presumably the main selection they've been under is the ability to grow in an incubator. And so here, what we can find is that by putting a new kind of selective pressure on them, we can get a change in cell behavior over just a fairly small number of rounds of selection. Now, these cells are so much better at migrating than the original population. Hansu thought we needed to give them a name. And so he proposed that we name them after Florence Griffith Joyner, who's one of the fastest humans alive. So for the rest of the talk, I'm going to refer to these selected cells that are rapid migrators as FLOJO cells.
Julie, quick question. Have you profiled them in different ways, number 6 or number 5 population?
Yes.
Are they just behaviorally different, or they also have genetic?
I am just about to show you. All right, I'm not going to show you all of the experiments, but I'll show you a smattering. So the first thing we did when we got these FLOJO cell population was to put them through different kinds of motility assays. So they were originally selected by going through this Transwell assay to recognize a chemoattractant called SDF1 alpha. It turns out those same FLOJO cells that are shown here in pink, they're also better at migrating in response to different kinds of chemokines. They're even better at this Transwell assay in response to just serum, to presence or absence of serum. So it's not just that they're better at detecting the chemoattractant they were selected on, they seem to be better at detecting all chemoattractants. More importantly, we also are able to do a huge number of different kinds of quantitative motility assays. So these cells, they love to crawl. We can make them crawl on glass cover slips in 2 dimensions. We can make them crawl through 3-dimensional collagen gels. We can put them in the presence of different kinds of directional signals. So for example, these cells like to respond to an electric field. So we can put them in an electric field and measure how well they migrate in that context. If you want to know more details about electric field migration, please ask me afterwards. It's a great story. But for this purpose, I just want to show you that when we now look at the FLOJO cells, which remember were selected on this assay of crawling through holes in response to a chemoattractant, if we now put them in an electric field, basically they're better at everything we can measure. So they move, so they move faster than the control population. They move in a more directed way, just intrinsically, they move straighter than the control population. And when you put them under a directional signal, they're much more responsive, much more efficient at responding to the electrical signal. So these FLOJO cells really are the best athletes. They're just better at everything having to do with migration.
Does the non-directional diffusivity of the effective diffusivity of the cells?
Say again?
Does the non-directional effective diffusivity of the cells also go up?
Yes, yes, yes. And I didn't put all the non-directed data. I didn't put in all the 2D versus 3D, but we did all of those measurements. And basically, we've never made a measurement where the FLOJO cells aren't better. Yeah?
Like 2.5 months back, there was a paper talking
Could you
about-
speak a little more loudly?
I remember like a few months back, there was a paper in Cell about galvan in TMEM 150
Yeah.
controlling the directed cell migration.
Yeah.
Do you also see elevated levels of that particular thing in your selected FLOJO cells?
We do not, interestingly, but we don't.
Try to repeat questions.
Oh, sorry.
Your
Yes.
mic, then it's a bit.
Okay, so the question was about a protein called galvanin that was discovered by my postdoc Nathan Beliveau in neutrophils that's responsible for detecting the electric field. And so the question is whether we see upregulation of galvanin, and we don't particularly. We also don't have any evidence that the responsiveness is under normal circumstances is tied to the galvanin expression level, but that's, I think, sort of another point. Okay, anyway, these cells are just better at everything. So, so of course the question is, what's different about them? So the first thing we did was just bulk RNA sequencing. Hansu basically did the selection with 3 independent biological replicates, taking things through in 3 parallel pathways, and then did bulk RNA sequencing relevant to this aging control, the proliferation burden control. And found, several hundred genes that are differentially regulated. And if we look at any individual genes, the top 2 are ones that increase over selection. As the bars shown in red, you can see they go up pretty steadily from generation 4, generation 5, generation 6, generation 7. And the bottom ones show 2 examples of genes that are downregulated through the process of selection. There too, we seem to see this gradual decrease in expression level. Now, of course, just looking at the bulk RNA sequence, we don't know whether that's because all cells are increasing or decreasing their expression, or whether it's a subset of cells that are becoming more enriched. So we followed this up with single cell RNA sequencing. And here we've got now our original population, just as they came from the ATCC, is actually by single cell RNA sequencing quite distinct from both the FLOJO cells, which are the rapidly moving cells, and the control cells, which are the ones that have had the same proliferation burden. And just looking at the overall distribution of expression, you can see those 3 groups actually separate quite nicely. But if we break them out and look, for example, just at the original cells, the flojo cells, the fast-moving ones, are mostly in this big branch called 0. But in the original population, we did have a small fraction that were actually in that same part of the UMAP plot, meaning they had fairly similar gene expression to begin with. So if we take our selected cells, about 90% of them cluster in that major group. And from the original cells, that was less than 1% of the population. So this is at least consistent with the idea that what we're actually doing here is selecting a pre-existing population rather than necessarily inducing alterations over time.
You didn't have to barcode your cells before the experiment.
Barcoding experiments are underway. We just sent off the sequencing. Yeah.
How long-term stable are these cells? If you would just grow them without the suction pressure for a month,
yeah,
be reverting back?
That's a great question. We've only explored that a little bit, basically by taking the selected cells and then doing various different dilutions to force them to proliferate. And we have to do a pretty serious dilution in order to actually get them to start dropping their flojo, their migration strength. We don't have specific numbers on how long it takes, but it's looking to me like it's heritable probably at least over the order of about 10 generations and probably more. We haven't done that carefully yet, though. Yeah.
Is your thought for these cells that the contribution is coming from something that's non-genetic and heritable in a non-genetic way, or do you think there's actually a genetic basis?
Don't know.
Okay.
Don't know. Could be either way. Yeah.
Have you tried this with any other cell types?
No, actually, so a little bit of backstory. The reason that we're using these EL4 cell types instead of any of the other migratory cells we've used before is because this cell line is unique in being extremely rapidly motile and also proliferative. So for example, neutrophil-like cell lines we've used extensively, but those guys you have to differentiate. And so you can do genetic manipulations, but then they get terminally differentiated. You have one shot at an experiment. And so in order to do this thing with rounds of selection, this is the only cell type that we know that has both the criterion of being rapidly moving. So it's not like a fibroblast or whatever, but like rapidly moving and also proliferative. So this is the only one we've done it in so far. Yeah.
How stable the genomes are in these cells?
No, that's a great question. We don't know and haven't been able to find any literature about it either.
Can you look at just, use pretty out of the box methods like for CNV to just tell if there are giant chromosomal alterations that happen between 0 and cluster 0 and 3, because these are huge differences.
Yeah. In principle, we absolutely could. We just haven't done it yet. Yeah.
Did the stiffnesses of the cells change before and after the selection?
We haven't measured stiffness. We did measure adhesivity, so their ability to adhere to a plastic substrate, and they are substantially more adhesive after the selection. Stiffness would be a good thing to measure too, though. I bet you $1 it does change. Yeah.
Might have mentioned, but morphologically, they do look flojo.
Morphologically, they look the same. We can't tell the difference by looking at it.
Have you tried just treating with like an active demethylating agent, like 5-base?
Oh, we've tried all of that. Yeah. And no, we can't disrupt it in any obvious way. Either we kill the cells or else they still remember their state. Yeah.
At the receptor levels, have you checked if the receptors are increasing over generation for the cytokine?
Yes, for all the cytokines, no change in receptor level.
Oh, really?
Yeah. Yep.
So you're probably going to get to this, but if you increase expression of some of those integrins.
Thank you. What a nice segue. So many of the genes were upregulated, as you saw, and of course it's very appealing that cells are going to be able to do the motility better if they upregulate things that are associated with. So between the single cell RNA sequencing data and the bulk RNA sequencing data, we have a nice list of what all the things are that seem to change. And so Hansu took basically the top 20 or so and made single gene overexpression lines of those and then tested them individually. Up here, we're looking at the data in the Transwell assay. This is the control. So about 1% of the cells go through the membrane on the first round of selection. And of the things that show up as being most significant in the plot, when we do single cells, the very best one only gets up to about 6%, whereas remember the flojo cells, the fully selected ones get up to about 50%. So none of these things are able to explain more than a tiny fraction of the difference between the starting population and the flojo cells. And again, we can do many different assays. I just heard the timer, so I'm not going to go through this in detail, but
1 minute.
Okay, cool. So I hit some highlights basically by looking at single cell or single gene overexpression. We can separate out ability to migrate in 2D from ability to migrate in 3D from directionality. And we now have single genes that actually correlate with only one of those things, although the flojo cells are better at all of them. So that's where we are in terms of this ongoing experimental project. I would love to follow up with some of the other characterization that people have been suggesting. As I mentioned, the barcoding experiments in the style of Yogesh are currently underway. So the sort of provocative set of things I want to leave you with is, how to think about cell states and cell state transitions in this kind of context, where we're dealing with an immortalized cell line. They just came from the ATCC. They're allegedly a single population, and yet we see there's all this sort of heterogeneity in them. And when we select on function, on cellular function, which is highly multifactorial, we see that these variations are both persistent and they're also heritable, at least over some sort of order 10 generations. So what I'd like to put to this group is, given that this is, natural variation that you see even in the simplest conceivable kind of mammalian cell population, which is an immortalized cell line that's been living in the incubator for decades, even there, we clearly see a continuum of cell behaviors, cell gene expression, all that kind of thing. And it's not clear to me how we go about describing cell state when we have something like the FLOJO cells, which is the original population, are easy to distinguish from each other by any of a number of criteria. And yet, in some sense, they're all still in the same large state, in the same large attractor basin of being an EL4 lymphoma cell. So how do we rigorously define cell states when they exist along a continuum? And then also, and this gets back to Herbie's question, is how persistent does the variation have to be in order for us to consider this as really a cell state that the cells are trying to maintain? And is a few generations long enough? If a cell changes its behavior over a short period of time but then reverts, can that be considered a cell state? I think we're very much at the early days of even just trying to define these things. In this particular case, we selected cells based on function that we could select, we could measure. And it so happens that this particular function of finding these FLOJO, best athlete cells, are well correlated with a lot of other cellular observables. And in particular, we took advantage of the fact that it's correlated with changes in gene expression profile. Don't know if that's causative or not, but at least we can use it as a marker. Is that always going to be the case? I mean, cell behavior, cell function is so complex. It involves so many multifactorial things. It's not clear if we're always going to be able to define these variations in behavioral states with respect to something that's easy to quantify, like molecular census. And then in this case, I would say if you made me bet, I would bet we're doing selection here rather than actual induction of change. But we'll know that when we're done with the barcoding experiment. And that raises the possibility of whether behavioral states could be induced. So if we started with just the non-FLOJO cells from the original population, could we still get them to come out after sufficient rounds of selection? And in this particular context, of course, it's really interesting to see that there's so many different motility-related factors that are all coordinately regulated, although it's not known that they share common transcriptional machinery. So how that common regulation is done is also of interest, whether that has something to do with chromatin state or potentially even with genome duplications or deletions in part of the genome. We just don't know yet. But I think it's worth thinking about because there's also, as I showed you in the very first slide, really nice experimental evidence for extreme functional heterogeneity in primary T cells that actually come from an animal. So understanding how these things are related, I think, could give us more insight also into T cell biology. So as I mentioned, this is almost entirely the work of one graduate student, Han-Soo Kim, with a little bit of help from Nathan Beliveau, former postdoc, and Sage Allen. And thank you all for listening.
Come up. Where's Lior? Yes, as the next speaker. Maybe I'll ask you a quick question. Your other population, the parental population, was so far away.
Yeah.
If you did the same experiment 10 times with single cell bottleneck individual clones, do you think you will get the same genes? Like I personally believe, genes are not necessarily always the true labels of the phenotype. You could probably get a bunch of different ways to get FLOJO.
Yeah.
What are your thoughts on it?
Yeah. So for technical reasons, it's very hard to actually isolate single clones. If you put an individual EL4 cell in a multi-wall plate, it just dies. It needs to have its friends around. So we have to do it with a feeder layer. So we have to have labeled versus unlabeled cells and things like that. We just haven't actually done that. I don't know. I can't wait to find
Is it there's a canalized way to become FLOJO, or there are 5 different of the
we've done between Han-Soo and Sage, who is a rotation student. They've gone now through the selection. I'm going to say something like 8 or 12 times, just completely independently from the starting population. And the guys we get at the end always are very similar phenotypically.
Okay.
Now, whether that's because of catalyzation or whether that's because of selection of a
Wonderful,
persistent pre-existing population, we
But
don't
when
know.
you say phenotypically, you're including the molecular phenotype?
Yeah, yeah.
Okay.
And we haven't done the extensive molecular phenotyping on everything else, but we have a couple of markers that are easy to look at. Yeah.
You put all of those together in one cell, like take all your hits and overexpress them.
Yeah, so Hansu started making pairwise ones and we actually just stopped doing that because that way lies madness. There's no way we can.
Also, like your setup seems like it'd be really amenable to like a functional screen too, right? Take EL4 cells, hit them with like a CRISPR-A, CRISPR-I.
Yeah, absolutely. Like I said, this was a control experiment to begin with, and it was just for that. So we've done the CRISPR-I survey in HL60s in neutrophil-like cells where we only get one shot, and we got a lot of cool stuff out of that. Originally, we wanted to see if these guys would show, a similar suite of regulators as the neutrophils. But we figured before we did that, we should just first see, what they do. Are they stable if we just put them through the filter? And it turns out the answer is no. So we can't yet do the actual genetic selection, I think, until we understand the parameters here. But that was the original idea.
Okay, last question, and then we'll get started.
Can you tell the difference between selection on pre-existing heterogeneity versus the gene regulatory response and response to the challenge that you're giving to the child?
We can't right now. I think actually the best single experiment for determining if it's pre-existing heterogeneity is the barcoding approach. And so what Hansu did for that was he has a barcoder library, went through rounds of selection, I think 6 different lines parallel, and then 3 separate lines for the proliferation. So it's off being sequenced. I should be able to let you know more.
Thank you.
Thanks.